A sea slug eats a jellyfish and comes out armed. It fits on your thumbnail. Smaller than a paper clip. Electric blue and orange, serrata bristling from its back like a tiny crown of fingers. This is an earlid nudibranch. And it just consumed something with a nervous system designed to fire venom into anything that touches it. The jellyfish tentacle contains nematocysts. Pressurized capsules, each one a coiled harpoon wound tighter than any human-engineered spring at that scale. Under electron microscopy, first captured clearly in 1972, they sit like loaded guns. One wrong chemical signal, and they discharge in under 3 milliseconds. The nudibranch ate them anyway. Not digested, not neutralized, transported. Rudolf Berg described the serrata structures in 1890, but it took nearly a century before anyone understood what they were actually carrying. In 1984, researcher R.N. Mariscal at Florida State University published the definitive proof in Marine Biology. His words were exact. The nematocysts are transported through the digestive gland tubules and into the cnidosac at the tip of each serous without discharging. Without discharging. Through living tissue. Intact. Terence Gosliner at the California Academy of Sciences cataloged over 50 earlid nudibranch species doing exactly this by 1994. 50 species. Separate evolutionary lineages, same impossible trick. The stolen weapons end up in small swollen pouches at the tips of the serrata, waiting. When a small fish makes contact, it recoils immediately. The nudibranch does not move. The behavior has a name: kleptocnidae, stolen stingers. But here is what the research has never answered. The nematocysts remain functional inside nudibranch tissue for weeks, completely severed from the cnidarian nervous system that originally controlled them. Something suppresses the firing trigger during transit through the gut. Something keeps a loaded weapon from going off inside the animal carrying it. As of the most recent published study on the mechanism, that suppression signal has not been identified. By the end of this video, we will reach exactly that question. The specific mechanism researchers have studied for decades, the chemical signal that should not work, and why it still has no complete explanation. The first answer was clean. Elegant, even. The nudibranch has a chemically resistant gut lining. The nematocysts pass through. They never fire. Case closed. Except the evidence wouldn't stay closed. Nematocysts don't need much to discharge. They respond to two things: chemical signals and mechanical pressure. Both exist inside any functioning digestive tract. Stomach acids. Peristaltic movement. The physical compression of tissue passing through a narrow tube. Any one of those should be enough. Hans-Jürgen Mariscal at Florida State University proved the problem in 1984. In Marine Biology, Volume 81, he documented nematocysts arriving at the serrata tips, the skin structures at the nudibranch's surface, completely intact, capsule wall unbroken, coil still wound, still loaded, still armed, not neutralized, not degraded, fully functional. That is not what a gut lining does. A resistant lining protects the animal from the weapon. It does not deliver the weapon. Craig Greenwood at the University of California Davis proposed a refinement in 2003. Mucus. The nudibranch might coat the nematocysts in a chemical secretion that suppresses the discharge threshold, essentially a molecular dampener applied during ingestion. Published in the Journal of Experimental Marine Biology and Ecology. A real finding, a real mechanism. But it only moved the problem one step back. Conor Caffrey at the National University of Ireland, Galway, confirmed in 2006 that nematocysts recovered from Eolidia papillosa serrata were not just structurally intact. They were capable of discharge. Functional. Weapons, not relics. The mucus, if it existed, had worn off. Or it was never the whole answer. So the nematocysts arrive loaded. The gut environment should trigger them. The mucus hypothesis slows the question down, but doesn't stop it. Which leaves one question standing. If nothing neutralized them, if they traveled through living tissue, fully armed and arrived ready to fire, what stopped them from firing in transit? The nudibranch swallows the nematocyst. The nematocyst does not fire. That much was settled. But the question nobody could answer. Not in 1972, not in 1984, not in 2003, was why. In 2004, a research team led by Paulette Greenwood published findings in the Proceedings of the Royal Society, confirming the suppression was real, reproducible, and measurable. The nematocysts arrived at the cnidosac intact. Still loaded. Still viable. The transit had not degraded them. Something had simply held them quiet. That something has no name. The nudibranch routes nematocysts away from the main digestive chemistry, through the gut wall into the serrata tips, without once triggering the mechanical threshold that would fire them. Greenwood confirmed the pathway exists. The molecular signal that keeps the weapon silent during that entire journey remains unidentified. In 2009, Greenwood wrote in Toxicon, the mechanism by which eolids prevent nematocyst discharge during ingestion and transport remains one of the most intriguing unsolved problems in marine biology. When isolated nematocysts from a cnidosac are removed and placed into a chemically identical gut environment, they fire immediately. Same chemistry, different context. Something in the living tissue suppresses them, and science cannot isolate what that something is. Smaller than a paperclip, carrying weapons it stole, sorted, transported alive through its own body, and deployed on command, without a single misfired round. The nematocysts can remain functional inside nudibranch tissue for weeks, completely severed from the cnidarian nervous system that originally armed them. Terence Gossliner at the California Academy of Sciences cataloged over 50 eolid species doing this. 50 species. One unsolved mechanism. Demetra Andrikis at James Cook University traced cnidarian DNA through the nudibranch digestive pathway in 2012 using molecular techniques. The DNA confirmed transit. The mechanism that governs it remained outside the data. That is the gap, not a failure of science. The sharpest open question in invertebrate biology, filed here unanswered, exact.